Namaste friends, welcome you all for the biology session. In today's class, let us discuss about the chapter microbes in human welfare. The chapter mainly which deals about how microbes play a major role in obtaining a product. So microbes play a major role in obtaining household products and to obtain the industrial products. And along with that, these microbes are also very useful in the obtaining organic acids and enzymes, organic acids and some very important enzymes and chemicals certain chemicals. So these enzymes, which are generally some which are used as the organic acids, or you can say that some enzymes which will be used to treat some disease, right? And along with that, the microbes which will be used mainly to obtain organic acids, and along with that, generally which are also play a major role in the sewage treatment plants. So STPs in this chapter we're going to discuss about, and where in which we can say that these microbes are also acts as biocontrol agents. So these are very important the points, or you can say that the important things in this we're going to discuss about. And these microbes which will be there, right, they play a major role which helps in enhancing the fertility of the soil as biofertilizers. So let us take one by one and we can take some questions related to this. So first thing is that in household products, generally to obtain microbes in the household products, where we can say microbes in household products, where we can say it used to obtain long ago, right, from our ancestral, or I can say immemorial time, right? So small organisms which generally these can survive anywhere, right, anywhere on this earth. So that is what is it, the cosmopolitan, right? So these which can survive in extreme conditions, snow, thermal vents, highly acidic the habitats. So because of that, these which play a major role and which are also not only harmful to us, they are also the very useful to us, very much useful to us. So in this we're going to take only that how they are useful to us. So microbes belongs to a different group, right? So generally here, so one thing you have to keep it in mind, that these microbes which belongs to mainly the group monera, that is bacteria, and which are also belongs to fungi, right, and protozoa, right, fungi and protozoa, and along with that some microscopic plants. So these which are we will consider as the microbes. These microbes belongs to a group of organism: bacteria, fungi, protozoa and microscopic plants. And some which are there, that is we can say that the viruses, viroids and the viruses, viroids and prions, right? So viruses, viroids and prions, right, that is proteinaceous infectious agents, prions, right? So these are also included under microbes, where the genetic material will be varies, right? So first thing is that in the household products, that mainly which used to obtain dairy products. So dairy products, as we all know, that every day we're going to use the milk, curd, right, ghee, cheese, right, milk, curd, and then the cheese. So these are which will be used, and along with that we're also going to use yogurt. So in this, generally the lactic acid bacteria, which is very much important, this lactic acid bacteria which will be used to convert the milk into curd. So right, the small amount of the lab which will be added, generally we can say that the small amount of curd which is already the there, that generally which will be added to the fresh milk which convert into the curd. So milk will be converted into curd, right? So here lab which will be present in what is that small amount of curd. So that small amount of curd which will be used as a what is it, the starter, that is called as inoculum. That is called as inoculum. Generally we'll say that is a small amount of milk which is with the which is with the lab, that is where you can say that with the millions of lab, that is lactic acid bacteria. So curd is obtained, right, using generally here the one example for that, to obtain a curd, lactobacillus acidophilus will be used. Lactobacillus acidophilus, what is it, bacteria will be used. And to obtain what is it, the generally to obtain yogurt, here also we are going to use what is it, generally lactobacillus bulgaricus, and where we are also going to use what is it, the generally streptococcus thermophilus, what is it, the thermophilus, which will be used. So generally milk is curdled with the help of lab, that is mainly to obtain generally curd, and curd is heated to separate cheese from whey. So that is what we can say. Generally if you consider that cheese which will be there, that curd which is obtained, right, curd will be heated. So after eating that curd, so curd heated to separate cheese, right, to separate cheese from whey, from what is it, whey, EY, so WHEY, whey. So this is what you have to keep in mind. And what is that exactly, we can say milk, when lactobacillus so acidophilus will be used, so milk which will be converted into what is that, curd. So lactobacillus acidophilus, keep it in mind, that is also lab example for lab. So small amount of curd and along with that fresh milk when added, that is called as inoculum. And next, here they also used what is that, the obtain the household products like dosa, upma, that will be used, what is it, by mainly how it will going to obtain, that is by the fermented preparation of rice and black gram. So this is what you have to keep in mind. And here they are going to use to obtain idli upma, and this one they are going to use, right, streptococcus species and leuconostoc, right, leuconostoc and streptococcus species. So next, and there they are going to use what is it, the bread. So other we have studied, that is a bread. So that is we can say in the bakery items, where you can say that Saccharomyces cerevisiae, that is Saccharomyces cerevisiae, Saccharomyces cerevisiae, Saccharomyces, what is that, Saccharomyces cerevisiae, which will be used to obtain what is that, bread, that is baker's yeast. So baker's yeast, so that is when it will be added with the baker's yeast, means Saccharomyces cerevisiae, when it is added with what is it, the flour, any what is it, you can say that the cereal flour which will be used, mainly we can say maida, will say, right. So that will be needed, that will be mixed with baker's yeast, and that will be warmed for few hours, and that is what which will be swell up, and that which will be baked, right. So when in which the fermentation will be takes place, carbon dioxide and ethyl alcohol which will be evaporated, generally, right. So that is making bread that makes bread porous when it is when you're going to bake it. That is, it leads to evaporation of these things, and bread becomes, what is it, the bread becomes porous, right? Porous and smooth, along with that smooth and soft. So this is what. And finally we're going to use, that is, earlier they used to obtain toddy. Toddy is what, actually fermented sap which is obtained from what is it, the palms, that is latex or latex sap or latex we can say. So that is obtained. And some which is say microbes which will be used to fermented fish and soya, for the fermentation of fish and soybean, and bamboo. Earlier people used to have food after treated with, what is it, treated 1 or 2 days with a fermentation process, and they used to have, right? So this is what bamboo shoots they were used to obtain food, fish, and after fermentation they used to have. And their very important thing, tempeh, what is that, tofu, sofu, tempeh, tofu and sofu, which will be used generally, right, as a food, right? These are the different types of food. Tempeh, tofu, sofu are the different types of food. These are the fermented food which is obtained from soybean, and, right, fermentation of, what is it, the soybean and along with wheat, right, we're going to obtain this food. So tempeh generally in Indonesia they're going to have, and tofu that is in, I think, Japan, right? So that is in Japan, and sofu that is in China. So these are all the products which are obtained by the fermentation of soybean and wheat. So next here, the major thing you have to keep it in mind that, so whenever we're going to obtain, there we're going to obtain the important question we can get that cheese, right? So cheese is partially degraded concentrate, that is partially degraded concentrate milk fat, right? That is concentrate milk fat and milk sugar, milk protein, milk, was it, fat and protein. So this is what you have to keep it in mind. So here keep it in mind that milk sugar and milk protein, when completely coagulates, that give curd, right? So milk sugar, that is a lactose, and protein, that is a casein, which will be completely, what is it, the degraded. It is partially degraded, which gives cheese. If it is completely degraded, that is curd. You have to keep it in mind. Next, so there are as we all know that when protein, that is casein, milk protein casein, which is degraded, which used to obtain, what is it, there are two different types of cheese, Swiss cheese. So characteristic taste and quality of the, what is it, the cheese is determined by which type of organisms, microorganisms used to obtain. So Swiss cheese, which will be with the characteristic feature, will be with large holes and generally what is it, the which is not slightly bitter, which is not bitter, then which will be obtained from the bacteria, right? Keep it in mind, Swiss cheese obtained from bacteria called as, what is that, Propionibacterium, what is that, Propionibacterium shermani. So this is the bacteria, what is it, which is used to obtain, what is it, Swiss cheese. So this is actually thermophilic bacteria, which is going to survive in, what is it, high temperature, thermophilic bacteria. And one more, that is the Roquefort cheese. So Roquefort cheese, which is obtained using, what is it, fungi, right? Directly they may ask, using which microbe, bacteria, that is Swiss cheese, that is Propionibacterium shermani, and Roquefort cheese, which is obtained using fungi, that is Penicillium roqueforti, right? That is Penicillium, right, roqueforti. So this is a one which will be used to obtain, what is it, recipe for cheese. They may ask direct question. Right? And one more, the additional here, it is not mentioned in the NCERT book. That is, we can take what is that camembert cheese. So this is also obtained using generally what is it, the fungi, that is Propionibacterium. In short form, we can write P. camemberti. Propionibacterium, what is it? Camemberti. So this will be used to obtain what is that? Cheese. So this is very very important. The characteristic feature of where you can say quality, right? So quality and the taste, characteristic taste depends on which microbe will be used, whether bacteria will be used or fungi will be used. So that is what you have to keep it in mind. So here note you have to keep it in mind that when generally milk sugar which will be treated, so first when milk treated, that is which is there in the milk sugar, that is lactose, milk sugar lactose, in the presence of lactobacillus. So this is a common thing you have to keep it in mind, right? General thing, that is converted into lactic acid. So that is what this lactic acid, when it what is it, the milk is treated at 40 degrees Celsius, right? So now this lactic acid which acts on milk protein, right? So milk protein, that is casein, milk protein, that is casein. When this casein which will be completely degraded, if it is completely degraded, we can say that is curd. If it is incompletely degraded, that which is going to obtain cheese. But we are going to use what is it, the different lactic acid bacteria, right? So this is what right. In case of to obtain yogurt, we are going to use Streptococcus thermoacidophilus, or you can say Lactobacillus bulgaricus, right? So this is what you have to keep it in mind. And cheese is partially degraded concentrate of milk fat. So here I have mentioned, so this is very very important, partially degraded. So you can, what is it, the just I will show that this is a partially degraded concentrate milk fat and protein, so this is what you have to keep it in mind. Next, microbes in industrial products. So here the second heading we can take, that how these microbes play a major role in industrial products. So to obtain that industrial products, so generally, so you have to keep it in mind that which will be used, mainly the industrial products, which includes what is it, the mainly the industrial products which are of two types, that is a primary microbial products and secondary microbial products, right? Which used to obtain generally enzymes and then amino acids, vitamins, and along with that antibiotics, alcohols, and organic acids. So if you consider that, mainly here we can mention that in the industries to obtain what is it, the alcohol and antibiotics, beverages and antibiotics. So industrial products mainly includes what is it, beverages and what is it, antibiotics. So in this we can take to obtain these beverages and antibiotics, generally they are going to use a large vessels. So those large vessels which are called as fermenters, so this is very very important. Large vessels which are used in the industries to obtain, they are called as fermenters. So in this, generally to obtain fermented beverages. So in this, let us take, that is how these beverages will be obtained. So first let us take the beverages. So beverages which includes, right, generally by using the yeast called as Saccharomyces cerevisiae, which are called as brewer's yeast. So yeast which is also called as baker's yeast in bakery, where we can say bakery foods will be obtained, and here Saccharomyces cerevisiae which is also called as brewer's yeast. Saccharomyces, what is it, cerevisiae, Saccharomyces cerevisiae. So this is which will be used added with other, what is it, the almost all the fermenting materials to obtain. So now that is Saccharomyces cerevisiae, which will be used right to ferment, right, malted cereals, and maybe fruits, right, and juices, cereals, fruits, and juices. What is it, mainly to produce, right, Saccharomyces cerevisiae used to ferment, right, ferment malted cereals, fruits, and juices to produce alcohol. To produce, that is what, alcohol, where you can say that ethanol. That is ethanol. So this you have to keep it in mind, like where they are going to use wine and beer to obtain wine and beer, mainly to, that is, they are going to obtain without distillation. So this question we can expect beverages like wine and beer without distillation, whereas whiskey, brandy, rum, right? Whereas whiskey, brandy, right, like rum, which will be obtained with distillation. With distillation. So these are the things you have to keep it in mind. So next here direct question we can explain. Barley malt will be used to obtain beer, whereas the cereals used to obtain whiskey, right? So some cereal malt which will be used to obtain, what is it, the whiskey. And along with that, generally if you're going to take that rum, which is using, what is it, molasses will be used, and brandy which will be obtained using, what is it, the generally where we can say that the using juices, fruit juice. So fruit juices will be used, right, to obtain brandy. So rum, molasses. So next, antibiotics. So what are these antibiotics under industrial and then industrial products? We can say that these are the antibiotics are the chemicals. Antibiotics are, we can say that the substances produced by microorganisms in low concentration. They produce in low concentration, mainly, what is it, inhibits. This is very very important. Inhibits the growth of microbes and then metabolic activity. Growth of microbes, and also inhibits the growth and metabolic activity of the microbes. They're going to, what is it, they affect mainly the activity of the pathogens, right? Microbes which acts as pathogens, directly here we can say microbes which acts as pathogens. So inhibits the metabolic activity, and without affecting, antibiotics, they're going to kill these pathogens, but without affecting host. Then only we can consider as antibiotics, that they'll produce in low concentration. They affects growth and metabolic activity of the microbes, that is, which acts as a pathogen, but without affecting the host. So best example, which is generally obtained by Alexander Fleming during Second World War, right? So that is the, what is it, penicillium. So penicillium, which is obtained using penicillin, that is, penicillin is an antibiotic which is obtained using Penicillium notatum. So this is the one which is used, right, mainly to obtain, that is for the first time, which is obtained using Alexander Fleming. So Alexander Fleming, right, for the first time who obtained, that is this in the 1928, right? So 1928. In this, so many, what is it, the antibiotics which we use, and later this commercially obtained, we can get this question, point to be noted. This is commercially which was obtained by Ernst Chain, that is Ernst Chain and Howard Florey. So these are the direct questions where we can expect in the CET. So commercially obtained, right, so extracted by the efforts of these two fellows, right? So later they started using, so keep in mind that mainly the bulk of the antibiotics which are obtained from, so antibiotics obtained from, this question also we can obtain, that is obtained from eubacterium, right, or eubacteria, not, what is it, the, right, that is the fungi, it is not Actinomycetes, that is Actinomycetes, right, that is the what we can say that the filamentous bacteria, and along with that fungi which will be used, right? So eubacterium, Actinomycetes, and fungi which will be used to obtain antibiotics. So antibiotics which are obtained from Eubacterium, actinomycete and fungi. So this is also we can expect this question. So next, so these which are used against what is it, the plague, whooping cough, diphtheria, leprosy, the many, that is like one best example which is used, that is what is it, the streptomycin. So streptomycin which will be used to treat what is it, the generally almost all which is treat very dreaded diseases. And in this we can take what is it, the third part which is obtained using, which is obtained microbes used to obtain organic acids, right? So here I will give one list. Already we know that this is one organic acids, that is microbes. So organic acids like citric acid which is obtained using what is that, aspergillus niger. So direct question we can expect, that is aspergillus niger. And then that is which is using what is it, the acetobacter aceti, that is obtained, used to obtain, that is acetic acid. So let me write what is it, the this is here, that is acetic acid, right? Acetic acid obtained using acetobacter aceti. So here you should keep in mind that this is fungi. They may ask citric acid obtained using bacteria or fungi, or what is it, the any other, this one, that is aspergillus. Name itself is going to give aspergillus, which belongs to fungi. Acetobacter aceti, that is bacterium, that is bacteria. And then what is it, the butyric acid. So butyric acid which is obtained using clostridium butylicum. So clostridium is it, butylicum. So clostridium butylicum used to obtain what is it, butyric acid, right? And then lactobacillus, we all know that generally lactic acid. So lactic acid obtained using lactobacillus. So there are many important things, and enzymes like what is it, the lipases which is obtained, right? So lipases mainly what is it, to remove what is it, the detergents in the laundry, that will be used what is it, to remove, right, used in detergents, that is in laundry, right? So mainly to remove oily stains. What is it, detergent which helps in removing what is it, the stains, that is mainly oily stains. So that is one which will be used, lipases. And pectinases and proteases, along with that we can also, one more example we can take, that is peptinase and proteases which will be used in what is it, the clarifying bottle juices, right? So that won't get thick, that will be what is it, the it always maintains a liquid what is it, the condition, right? So this is very important. And one more, that is here which is obtained streptokinase, that is streptokinase. So this streptokinase, right, which is obtained by streptococcus. So here we have to keep in mind that right, so only in this case, this is only acetic acid is the only one, that is a citric acid which is obtained using fungi. So all other organic acids here what we have studied, that is from the bacteria, right? So and then this is streptococcus, what is it, which will be used to obtain streptokinase, right? That is generally which is bacteria. Streptococcus which is bacteria. So what is the role of streptokinase, which will be used as clot buster? Streptokinase which will be used as clot buster, mainly to treat heart patients, or you can say that where to remove, mainly to treat the heart the patients, or we can say that the person who is suffering from to treat heart attack, or you can say that myocardial infarction. Myocardial, what is that? Infarction. To treat myocardial infarction. That is what clot buster, that is in blood vessels, mainly to what is that? The dissolve clot, used to treat or dissolve clot. Next, one more, that is which will be used active biochemical, or you can say bioactive molecules. Here we can say that these are the bioactive molecules which will be used, like example cyclosporin A, that will be used, what is it, the used as a immunosuppressive agent. When organ transplant will be done there to immunosuppressive agent, right? So that is very, very important. This is obtained from what is that? Trichoderma polysporum. So this which will be used, that is fungi. Direct question. So cyclosporin A, which has a immunosuppressive, used as a immunosuppressive agent, right? Mainly what is that? The used during organ transplantation, that is cyclosporin A, right? And then one more, that is statin. So statin is a one which is obtained from what is it? The Monoscus purpureus, right? So that which will be used mainly, which will be used as a agent which is used to lower the cholesterol content. So that is cholesterol lowering agent, which will be used as cholesterol lowering agent. So let us take related to this, and in the next part we can take that sewage treatment, and what is it, the sewage treatment, and which will be used mainly to treat the sewage, and we can take biofertilizers, and along with that how this will be used as a biofertilizers. So let us solve some question related to this, and in the next part we can take some other what is it, the concepts, right? So for this, study the following statement regarding the lactic acid. Just now we discussed which are used to convert milk into curd. So that is, they produce acid that coagulate, that is what we can say lactic acid. So that coagulates and partially digest the milk protein. So completely digest the milk sugar and partially digest so milk protein. So now here where we can say statement regarding lactic acid which are used in, so what is the thing, we have to select the correct answer. So they produce acids, that is correct, that coagulate and partially digest milk protein. So here we can say that mainly milk sugar, also milk completely digest. So during what is it, the process of conversion of milk into curd, milk protein, where we can say degraded completely, not partially, that is degraded. If it is partially, there we can say that is cheese. So that is why where we can say generally that statement we can take it as, so to produce a lactic acid and coagulates and digest milk protein. So this is also we can say correct, partially or completely degrade, but it coagulates, right? So that is also we can say it is correct, right? And next here, if it is partially the milk fat, if you're going to produce milk fat, here they are given digest milk protein, only milk protein. If it is milk protein and milk sugar we're going to give, that is a curd. Milk fat, that is cheese. So we can take this also, we can consider it a small amount of curd are added to a fresh milk, as in, right, as an inoculum, that is correct. So milk and curd, right, which is already obtained, that curd, and along with that fresh milk which will be added, which contains thousands of lab suitable temperature, that is what, at 40 degrees Celsius, and they're going to cool it, multiply and convert milk into curd. That is also correct. Conversion of milk into curd improves the nutritional quality, increasing vitamin B12. So this is also important. So correct. So whenever you notice, a lab may result in acidity in the stomach. No, this is wrong. So answer for that is, so first one is correct, second is correct, third is correct. This is wrong. 1, 2, 3. So that is C is the answer. Which of the following statement or statement regarding antibiotics are not correct? So antibiotics I already mentioned that. So which is generally used against the infectious or pathogenic agent. So antibiotics are attenuated microorganisms which are small concentration, can kill and retard the growth of other harmful microorganisms. So antibiotics are attenuated organisms with a small concentration, can kill and retard the growth of other harmful microorganisms. So generally these are the substances, right? They are not attenuated microorganisms, right? So that is incorrect. Penicillin was the first antibiotic was discovered by Alexander Fleming while working on bacterium Staphylococcus aureus. So that is also correct. The full potential of penicillin, right, as an effective antibiotic was established by Ernst Chain, and that is what we can say artificially obtained. This is also correct. Fleming, Chain and Florey were awarded Nobel Prize. This is also correct. So first is wrong. So only one is incorrect statement. So antibiotics are not attenuate microorganisms. Generally this will be used as a vaccine. That is the kind which is used as a vaccine. So which of the following antibiotics was extensively used to treat American soldiers during Second World War? That is which is obtained by Alexander Fleming. That is penicillin, which will be used as next. So identify the blank A, B, C, D in the following table and select the correct answer. So bacterium, so which is used to obtain. So generally we can say that the lactic acid, that is lactobacillus. So lactobacillus which will be used, that is where you can say that the A is lactobacillus, and fungus, that is cyclosporin A. So whenever we're going to use this cyclosporin, right, so Trichoderma polysporum. So that is Trichoderma polysporum. So B is, here we can get in these options we have the first one lactobacillus. So this we can't select. So out of these three we can select. Let us take next. So this is wrong, right, and these two options we can't select now in this Trichoderma. And C, Monoscus purpureus, that is a statin. So generally Monoscus purpureus, right, so which will be used, right, so whenever we're going to take, that is the yeast, Monoscus purpureus, that is also fungi that will be used to obtain the statin. And fungus Penicillium notatum, the commercial product obtained using, that is penicillin. So answer for this is, so here we can, this is not agaricus, that is mushroom. So answer is A. Answer is A. Right. Next. Which of the following antibiotic is not correctly matched with the source from which it is obtained? So penicillin. So penicillin which is obtained from Penicillium, the Penicillium chrysogenum. We can select this also. That is bacitracin. That is Bacillus licheniformis, and this is also correct, right? And then Gracefulvin, Penicillium graciofulvum, that is also correct. Streptomycin, Bacillus gracius. So generally this is a streptomycin, where I mentioned that this is an antibiotic which is obtained, Streptomycin gracius, not Bacillus, that is Streptomyces gracius. So that is used to obtain streptomycin. So D is the wrong, right? Next, Bacillus thuringiensis, right, that is a biocontrol agent. Strains have been used for designing level. So where we can say that generally this will be used as, right, which used to kill the insects, that is bioinsecticides, bioinsecticidal plants, to obtain bioinsecticidal plant. So which used to kill cotton bollworm, and you can say that the generally this Bacillus thuringiensis, right, which is obtained from generally whenever they are going to kill that bollworm, mainly that is where we can say that the cotton bollworm, right? So that is we can obtain here. Next, where you can say bioinsecticides. Bioinsecticides. That is correct. Next, probiotics are. So probiotics are generally cancer-inducing microbes. No, it is wrong. New kind of food allergens. No, live microbial food supplement. No, that is they which will be used as what is it? This safe antibiotics. Safe antibiotics. So sorry. So probiotics which will be there in the curd. That is microbial food supplement. Curd, or you can say that some other. These are not safe antibiotics. Sorry, wrong. This is also wrong. And C is live microbial food supplements or probiotics. And organic farming does not include. So organic farming, green manures, it includes chemical fertilizers, right? It won't include chemical fertilizers, farmer manure and compost. So it's correct. And this one is not includes. So this is B is the answer. And farmers have reported over 50% of higher yield of rice by using which of the following biofertilizers. So this in detail we can discuss. Just I'll give here. So biofertilizers which will be used generally to enhance the soil fertility. So nutrient quality of the soil like nitrogen, phosphorus and other nutrient supplement will be what is it? The enhance. So in case of nitrogen fixing, what is it? The cyanobacteria, right? That is a blue-green algae which will be there like Anabina, Nostoc and Asletoria. Same like that. Best example here we can say that which helps in generally which helps in nitrogen fixing cyanobacteria. That is we can say which helps in nitrogen what is that fixing. The best association is Azolla and Anabina. So one is Azolla and Anabina, but most commonly used this is right Anabina. So Azolla pinnata, which is a free floating freshwater form. So this is the one blue-green algae which is what is it? Free floating. What is it? The water form, right? Hence it will be used in what is it? The paddy field. That is we can say that the rice field, right? They are going to use that right, used in a what is it? Rice growth. Right, so this is very important in China mainly and other what is it, the countries, right? So this will be used, right? It won't affect any other what is it, the plants, but it is going to fix the atmospheric nitrogen. Azolla, right, coexists with rice. So Azolla pinnata which exists with rice and generally helps in improving what is it, the quality or growth. Most commonly used green manure in India. So that is what most commonly used. That is Crotalaria and then Sesbania and what is it? SYNOPSIS. So all these will be used as a green manure. So which of the following statement regarding baclo viruses? So these baclo viruses which are there we can also consider as generally these are also which will be used in what is it, the biocontrol agent. So baclo viruses which attack what is it, the insects and arthropods, what is it, almost all the what is it, arthropods, right, which kill insects and arthropods. So majority of this what is it, the baclo viruses belongs to a genus nucleopolyhedrovirus, belongs to a genus nucleopolyhedroviruses. So nucleopolyhedrovirus, right, no negative impact on plants. They won't affect any other what is it, the host plants, mammals, birds and fishes, but they are specific and narrow spectrum only, right, insecticidal application. They're going to kill insects. Right, the majority of bacloviruses used to biocontrol agents include genus nucleopolyhedrovirus. That is correct. And generally that is the infection with bacloviruses occur when susceptible host eat virus particle present on foliage and become dead. So in this we can say that is also correct. And these of they will going to spray them. So this baculoviruses, which will be used as generally, we can say that the crystal form, right, that will be used most widely used, what is it, the microbial pesticides, along with which is obtained, that is a using Bacillus thuringiensis. So Bt toxin, along with that, this also will be used, and these are important organic forming specific action on harmful insects without causing damage to beneficial insects as well as environment. So all of these are correct. So all of these are correct. So 11th one, D is the answer. So baculoviruses, which will be used, all these Bt cotton and this one will be used in integrated pest management. So that is where we can say which won't kill beneficial insects. So this is what we can say, integrated pest management. So integrated, what is it, pest management, right? So without affecting the environment and other host and good insects, we are going to kill only harmful insects. Unicellular symbiotic organisms improve yield of legumes by, so that is where we can say, that is, we mentioned that biofertilizers. So generally mycorrhizae, right, which will be associated with symbiotic association of fungus and with higher root plants, with the higher root plants, that is mycorrhizae. And same like that here we can say where exactly that will be used, right, some in the leguminous plants, right. So that is where we can say that which forms mainly symbiotic association, they helps in nitrogen fixing, that is where we can say that the rhizobium, which is generally association which mainly helps in nitrogen fixing and forms a root nodules, right, root nodules in higher root plants, that is what, in leguminous plants. So leguminous plants, which mainly includes, what is it, rhizobium, what is it, the leguminosarum, leguminosarum, right. So and along with that, like we can say that generally here the legume plants, that is what, pea plant, and what is it, the other, what is it, the, so rhizobium. Other examples, generally we can say meliloti. So other examples are also there, just leguminosarum we can, that mainly which helps in fixing atmospheric nitrogen, will not fix atmospheric nitrogen. So this is wrong. Fixing atmospheric nitrogen, colonizing roots of host plant, that is what, which forms a root nodules, correct. Inducing host plant to absorb more phosphorus, wrong. So mainly that is to absorb more phosphorus, that mycorrhiza which is going to help. So this is what you have to keep it in mind, mycorrhiza which helps in absorption of phosphorus. Mycorrhiza association between, what is it, the fungus and higher root plant, fungus and higher roots, right, higher root plants, higher plant roots, higher, what is it, the plants, roots of higher plants. So that is also wrong. And stimulating the host plant to become tolerant to drought, so that is also wrong. So 12th one, so here we can say B is the answer. So unicellular symbiotic organism, so that is the best example, rhizobium, nitrogen fixing, dash produced by the bacterium streptococcus and modified by a genetic engineering used as a clot buster for removing clots and blood vessels from the blood vessels of patient who have undergone myocardial infarction, that is leading to heart attack, myocardial infarction. So in this case already we have discussed, that is the one which used as a clot buster, that is streptokinase, which is obtained using streptococcus, right. Next, select the option and fill up the blanks. Dash are used as a detergent formulations and are helpful removing oil stains. So that is, we already mentioned that, that is lipase, right. So these lipases which will be obtained, and then ripen growing Penicillium roqueforti, that is what, roquefort cheese, that is roquefort cheese which will be obtained. And then dash are produced without distillation, whereas produced with distillation. So wine and beer, so wine and beer without distillation, whereas right here we can select the wine and beer. So just we can interchange this, the whiskey and brandy which will be obtained, right, so using distillation. distillation of fermented broth. So broth means you can say malt, or you can say fruit juices, or any other. This one dash antibiotic. So that is one penicillin. So penicillin is the one which is used to obtain, generally obtained by penicillium notatum, to treat against the bacterium. So kushrobe, that is leprosy, is also called as kushrobe. So answer for that 14th question is lipase, liquid for whiskey, rum, and this. Just we can go through that. Just we have to interchange this, right? And then penicillin, leprosy. So we can select, that is B is the answer. So next, read the following statement and select the correct option. So biocontrol refers to use of biological methods controlling plant disease and pest. So in this we can say that is where the statement 1 is correct. Right, let us take use of biocontrol measures will greatly reduce the dependence on toxic chemical pesticides and chemicals and pesticides. That is also correct. So in this case, both the statement 1 and 2 are correct. But what are those biocontrol, or biocontrol refers to biological methods? So what are those biological methods which used? There we are going to use biopesticides, right? And along with that we are going to use we're going to use some insecticide that is naturally we're going to kill, right? So that is what biocontrol agents. So which are refers to biological methods which control plant disease and pests. Mainly we're going to use biopesticides. Here they should explain biocontrol, use of biocontrol measures where it includes biopesticides, hence they are going to the control plant disease. Not only biopesticides, and along with that they are going to use the microbial pesticides, and where these are the natural agent. So use of biocontrol measures will greatly reduce our dependence on toxic chemicals. Both are correct, but second one is not correct explanation for. So this 15th one answer is B. So let us take further questions.